Okay, everybody, welcome to this week's Flow-MER Friday, part of the webinar series. Just as usual, I'm going to give a quick introduction to myself and to the project before we get going. Then I'll pass over to the main presenters for the day. So, before we go any further, I'd like to acknowledge the traditional owners of the land on which we meet today and of all of the country within the Murray-Darling Basin and beyond in Australia. I'd like to express my respect and acknowledgement for their elders, past, present and emerging, and dedicate myself to working in partnership to try and redress past wrongs and ensure a sustainable and productive future together. Who am I? I'm Ross Thompson. I'm Director of the Centre for Applied Water Science at the University of Canberra. And in this particular setting, the most relevant thing is that I'm co-lead with Emily Barbour from CSIRO, who's also on the line today, of the basin scale component of the Commonwealth Environmental Water Office Monitoring, Evaluation and Research Program, which we call Flow-MER. This is a project which relies really heavily on the hard work and intellect of a whole range of different partners. The overall program is led by CSIRO and UC, but we work closely with scientists from Charles Stuart University, UNE, Deakin University, Saudi in South Australia, New South Wales Government Fisheries, Arthur Rylah Institute down in Victoria, and of course our partner in presenting this today is the Australian River Restoration Centre. Just by way of general introduction to the Flow-MER program, it is the Monitoring, Evaluation and Research program that has 8 main components, 7 selected area programs, which the selected areas are shown on that map there that's on your screen. And that's where data is collected in comparable ways to assess the effect of individual watering events within individual subcatchments, with the intention of being able to scale up those insights to the whole of basin scale. The basin scale integration program, which is what we're mainly talking about in these webinars, takes the data from those individual areas and looks at scaling up to whole of basin scale, at scaling up to areas where we currently don't have monitoring. So that's the 8 components of the Flow-MER program. There are 2 components within that basin scale evaluation. There is the research component, and it arose and draws most heavily from the UCA program, the Environmental Watering Knowledge and Research Program, which finished in 2019 and transitioned across to the research component within Flow-MER. And then the evaluation component, which draws most heavily on the long-term intervention monitoring, or LTIM, projects that finished in mid-year this year and which has now transitioned across to Flow-MER. So Flow-MER, because of that history that we've had more than a year of working with the research component, we're really just beginning to move into the evaluation component. These initial webinars are most heavily based on the research work, but we would expect that evaluation will become more of a feature as we go a little bit further on. If you are interested in further detail about the program, feel free to look at Flow-MER, the website, which Sawan and Pat have worked really hard on, and I think it's something we can be really proud of. It tells some great stories about environmental water. It is updated really frequently, and so if it's your first point of call if you're interested in knowing what we're up to. If you're interested in some more of the detail, then available now, and now beautifully branded with the proper TU branding, is the evaluation and research plan. That lays out the way that all of the different components of this project connect together, and that's available through the TU website. If you want more information, do feel free to reach out to either Emily or myself. That's our contact details up there. We can happily talk to you about this project until you beg us to stop. The webinar program. So this is, I think, the 5th of our webinars, and the webinar program was set up really to facilitate sharing of knowledge in lots of different ways and between lots of different people and levels. So particularly to share the research programs as they emerge and get feedback from local water managers and local engagement officers who hold a real wealth of information at site-by-site scale. to share ideas and information between the researchers at the selected areas and the basin scale team. And of course, to share information between and across agencies, whether that be MDBA, the TU, or state agencies. These presentations are recorded, and so there's a library of presentations that you can find on the Flow-MER website. And that's a great opportunity if someone's missed a seminar or someone's new to your team and they're trying to get up to speed on what's going on in the space. They can go back and watch those webinars, and that should be a really useful stakeholder engagement tool moving forward. So to date, we've heard from Heather McGuinness about waterbirds, James Hitchcock about food webs and energetics, Joe Bennett about refugia, and last week we heard from Brad Mogridge from UC and Emma Woodward from CSIRO about the Indigenous engagement component of the Flow-MER program. Today's seminar is an assortment of fish biologists. I'm going to describe it as that. They're going to introduce themselves and their affiliations as they start. And they're going to be talking about this really large basin scale component of fish projects that look at population biology and movement in particular across the whole of basin. So I'm sure you're going to enjoy it. I'm going to hand over now to the first of our assortment of fish biologists. This is Jason Thiem, and I hope you enjoy the seminar.
Thanks, Ross. Just bear with me. Thanks for the introduction. I think we've been called worse than an assortment of fish biologists before, so we'll see how we go. As Ross mentioned, Jason Thiem here from DPI Fisheries based at Narrandera. I'll be followed by Brenton Zampatti out of CSIRO in Adelaide, and then finally Charles Todd from the Arthur Rylah Institute in Victoria is going to wrap up. Ross has done a great job of introducing the Flow-MER project, and really I guess what I wanted to emphasise with this slide is that Brenton, Charles and myself are going to be talking about the fish research projects, and this is broken into, you'll hear us talk about F1, so fish population models that Charles actually is going to finish our session with. But firstly, Brenton and I are going to start with fish movement and population dynamics. Now, this is the assortment of fish biologists that Ross introduced earlier. So while the project team is large, as you watch and listen to this series of talks, you'll realise that we're drawing on large data sets collected for over many years from numerous regions within the Murray-Darling Basin, and that Flow-MER provides us with the platform to integrate these data sets to address new questions centred around the flow ecology of freshwater fish in the basin. So just to go into F2, which we're talking about flow movement and fish population dynamics. We just wanted to start with a brief introduction in terms of why fish move. So movement and migration are essential life history components for fish. They occur for the purposes of foraging, dispersal, reproduction, and even escape from adverse conditions. So they occur over a range of spatial and temporal scales. We see daily movement patterns in relation to home range use, driven by food and shelter requirements. The redistribution of fish throughout the landscape. So for this, we're talking about dispersal and key components of immigration and emigration. And obviously an emotive one is the obligate directional spawning migrations. And I guess something that we all think about is these spawning migrations of Pacific salmon in the Pacific Northwest of North America as they move from the ocean up into fresh waters and home to their natal streams to spawn. But we've actually got our own anadromous species, the pouched lamprey in the Murray-Darling Basin as well. So a range of these movements are regularly linked to flow, particularly those associated with dispersal and reproduction. So what we know is that movement ultimately influences the population structure in fish, and this is really what Charles is going to be talking about with F1. So understanding the spatial and temporal scale of movements is essential to maintain and enhance these populations. And understanding this is essential also to inform the appropriate management levers to maintain and enhance these populations. So while the primary focus of this research project and this series of talks is on flow management, we see fish passage and a number of other complementary measures as essential to restoring fish populations So you'll see in the figure in the top right there a map of the Murray-Darling Basin and all the barriers to fish movement mapped onto that. For some reason, we've actually left South Australia out of that one. But you'll see the idea there. And so we're also interested in habitat restoration. You know, other management levers include screens on pumps and fish stocking as well, but we're going to focus on flow management through this project. So in terms of our questions for F2, what we're interested in is event-based movement responses of fish and similarities between individual species to determine if these are repeatable, to standardise flows among species and regions. So I'm going to be talking and addressing part A of this research question. And part B is going to be addressed in Brenton's talk, where he's focused on the role of river hydrology in driving emigration and immigration rates of fish in the Murray-Darling Basin rivers, and how this varies by life history stage and species. So overall, our objectives are to refine our understanding of the role of river flows in driving movements for key species, and to use this information to inform flow management, both in monitored and unmonitored regions. Ultimately, what we want to understand is how flows associated with movements that influence population structure and provide these direct inputs into the fish population models that are being incorporated into F1. So to understand event-based movement responses to flow for fish, what we typically do is pick this gorgeous stretch of river here. This is an example from the Edward-Wookul River system. This is a common technique that we use to tag and track freshwater fish in the Murray-Darling Basin, where we actually capture a fish, put it under an anaesthetic and surgically implant an acoustic tag into its stomach cavity. So here you can see a photograph of an acoustic tag there next to the coin. There's quite a small tag that can contain a battery and basically emit a signal through the water that's decoded by the acoustic receiver that you see on the left. Each tag has a unique ID code, and when a fish gets within detection range, around 200 to 300 metres of that submerged acoustic receiver, we'll get a date and timestamp and a unique fish identification number. What we can then do, and this is a map of the Edward-Wookul River system in the southern Murray-Darling Basin that connects with the Murray. And so what we can do is we can place these submerged receivers or hydrophones at fixed locations or known distances throughout the river system. And as a fish moves past these individual receivers, we get information on which fish are in that region and how they're moving through that system. And we can actually recreate their movement history and calculate metrics, including the distance that they move through the landscape. Now, this is actually an animation that I was having trouble with this morning. So what I'm going to do is just share a new screen, and I've actually created a video of that. So bear with me for a second. Charles, nod if you can see that. Perfect. Okay, so what we've got here is an example of freshwater catfish moving through the Guarda River system. Thanks to Gavin Butler for this data. Each of the dots on that map actually represent submerged hydrophones. And what we see is the different colours and the squiggly lines moving through are different individuals moving in response to the hydrograph. We've got Guarda River flows in the top panel and Mihai River flows in the bottom panel. And you can see the river goes up, the fish move through that landscape, and we're quite good at visualising how fish respond to these flows at an individual level. Just going to go back to my talk now. And so when we move past that individual visualization, we start to get an idea of, or what we can do is calculate the distances fish move by knowing how far apart our individual acoustic receivers are. So here's another example from the Edward-Wookul River system. The graph that you see on the left there actually contains 3 different species: golden perch on the top panel, Murray cod in the middle, and silver perch on the bottom. We've got data plotted over time as fish move through the landscape with different coloured lines, purely just indicating different tagged individuals within this system. And this is just a way of demonstrating of how we can start to visualise that data and move from that sort of fairly descriptive movement example that we generated from the Guarda into quantifying the actual movement metrics of individual fish through that landscape over time. We can then aggregate this data at the entire sample level. So here you can see quite a complex plot, but I guess the simple explanation for it is we've got different panels going from top to bottom, indicating different years that we tracked fish through the Edward Waugh Cool system. We've got our top, middle and bottom panels indicating individual species, and we've aggregated our data at seasonal levels, so spring, summer, autumn and winter. And what we've done is we're looking at population or tagged population trends now in terms of distances moved. And from this information, we can start to see that spring is quite an important time for our reproductively mature individuals to be moving through the landscape. And in that particular example in 2016 that you see here, we get a lot of individuals moving quite large distances, which was in response to large flows and overbank floods in that system. So this information provides us with a snapshot of movement over a short period in a fish's life. This example's from Wayne Costa's work in the Goulburn River system. And so what we can do then is we can overlay individual movement plots and start to aggregate our data and match it up with our flow data. So what we see here is a golden perch in this plot that's actually moving. These individual dots on that graph indicate a fish movement downstream and then upstream in relation to flows over time. So we've got in periods of environmental flow releases. Here we've got our hydrograph of the blue lines indicating the overall flows within the system, and basically our river temperatures with responding to seasonal changes. And so once we aggregate that data up, we can produce these statistical models that give us confidence in our tagged population trends in terms of probability of fish movement in relation to time of year. In the top panel there, previous flow conditions that they experience within the Goulburn River system, and then movement responses or event-based responses to flows on the day or the 5-day period that they actually were recorded moving. So for those that have actually been hopefully paying attention, you will have seen that I've provided examples from the Guada in the northern basin, the Goulburn and the Edward Waugh Cool in the southern basin. And so I guess all three examples are those of LTIM funded projects in the last round of 5 years of monitoring fish movement responses to flow. But what we know through our collaborative networks is that we've got a bunch of researchers spread throughout all 4 basin states that are using the same technology in terms of acoustic tracking and quite often similar species to basically monitor fish movements in their own patch. And so what we realise once we overlay all of these different components on a map is that we're all doing quite similar things and often detecting each other's fish moving amongst individual acoustic receiver networks. So this really provided the impetus for us to approach the MDBA and establish an acoustic telemetry network across the entire Murray-Darling Basin. And you can see that last lot of dots that I put on that map actually joins up the individual acoustic receiver networks from different selected areas or different individual projects. And basically enables us to examine how we might start to link up some of these individual fish movements across different regions. So I guess to get to the nuts and bolts of what F2 and this component is going to do and what we're planning on is using integrated movement data from multiple species and locations throughout the Murray-Darling Basin, representing all basin states. And rather than examining movement responses to megalitres a day in the Edward Waugh Cool or the Goulburn, we're actually looking at movement responses to standardised flows among regions and to determine whether this is actually repeatable. So do golden perch move in the same way in the Goulburn or the Edward Waugh Cool or the Guada River system in response to bank full flows, not megalitres a day, so that we can actually, some of this information is transferable to unmonitored regions. So to come up with basically a set of data-driven rules for each species. And this was really a conversation that we started in LTIM. The figure that I've put up in the bottom right-hand panel there is actually some work Rick Stoffel's progressed in relation to the probability of fish spawning and using standardised flows from 3 different regions where we were collecting information on egg captures and probability of spawning in golden perch. So we're looking at doing that using fish movement data sets. And this component's really focused on event-based outcomes. So we're looking at daily, weekly, and monthly movement responses to corresponding flow events and moving away from individual megalitres a day and volumes to standardised flow metrics that are transferable to other regions. Just going to move on to Brenton now.
Okay, my turn, Jace. Thank you. I will just share my screen, everyone. Bear with me for a minute. Right, Jace, can you nod if you can see my full screen? Terrific. Okay, thanks very much. Hello to everyone. It is strange to be giving a seminar to 50 or 60 people and not to be able to see any of your faces or feedback. But I'm just going to imagine that you're smiling at me and listening intently. But the reality is you're probably checking emails and drinking cafe lattes. Anyway, Jace has just given a good overview of part A of our flow and movement project for MER. I'm going to be talking about part B here, where we're really looking at scaling up the work that Jace is talking about. So Jason was talking about using electronic tags to essentially look at fish movement at the site or reef scale at intra-annual timescales. And in part B of this project, we'd like to scale this work up to look at fish movement across a fish's lifetime and across river systems or along large river systems. So large spatial and large temporal scales. And these types of movements across a fish's lifetime are the type that may ultimately influence the structure of regional populations. So the abundance of fish in a particular location or the age structure of fish in a particular location. To do this, we've got to use a technique that enables us to look at fish movement across the fish's life. And for the two species we're interested in, which is golden perch and Murray cod, this lifetime might be about up to 25, 26 years for a golden perch. And for Murray cod, it might be double that, nearly 50 years. So we're talking about long time scales. But if we can know when a fish moves and where it's moving, we can ultimately associate environmental variables with those movements. And what we're particularly interested in here is flow. So Jace was talking about using acoustic tags and receivers to track fish movement almost in real time. But for a fish that lives for 20 or 30 or 40 years, we can't really use acoustic tags and acoustic receivers. So what we need to do is look for a method potentially to look retrospectively at fish's movement across their lives. And to do this, we're pretty fortunate because each fish is born with an otolith or an ear stone. And this is essentially like a flight recorder in the fish's head. And I'll explain what I mean by that. So up here on the left-hand side is an otolith from a golden perch. And this is a cross-section through that otolith. Now, some of you I know are familiar with otoliths or ear stones, but I'll just give you a bit of background. This is a calcium carbonate structure that's formed when a fish is born and continues growing throughout a fish's life. And you get daily or seasonal variation in growth. And so you get rings formed on the otolith when you get this variation. So when a fish is very young, we can actually count the daily rings on an otolith to determine how old it is in days. But on this otolith here, which is a golden perch otolith, which is about 11 or 12 years old, you can see the annual rings or the annual banding on that otolith. So we can determine how old a fish is. One of the other nifty things about otoliths is as they're growing, as the fish grows, they incorporate aspects of the water chemistry that's surrounding the fish. So if a fish moves between water bodies with different chemistry, those chemical signatures from those water bodies are actually integrated into the otolith. And so the otolith provides a time series of the water chemistry that's actually surrounding the fish. So potentially we can use the otolith to determine where a fish was and when it was there. And if we can do that, again, we can associate environmental variables. But for this technique to work, we first need to have an idea of water chemistry and be able to differentiate water chemistry between catchments. And if you look at this picture on the left here, this is the junction of the Murray and the Darling rivers. And you can see that there's probably quite large differences in water chemistry there. But we need to go to a finer scale than that if we're interested in fish movement throughout the Murray-Darling Basin. One of the candidate elements that we used quite a few years ago to determine whether there was differences in water chemistry between subcatchments of the basin was strontium isotopes. And the good thing about strontium isotopes is that strontium isotope ratio in water is an artifact of catchment geology. So if different subcatchments have different catchment geology, they potentially have different water strontium isotope ratios. So we knew we had differences between the Murray and the Darling. We needed to go to a finer scale. And we were able to do that through the EUCA project, which was really a precursor to this MER program. And through the MDB EUCA project, we were able to develop a strontium isotope isoscape for the Murray-Darling Basin. And for about 8 years, we'd been collecting strontium water samples for the southern basin, but the EUCA project gave us an opportunity to work with a whole lot of colleagues across the basin to integrate water samples from the north as well. And Ben Fanson from the Arthur Eiler Institute developed a model which is able to represent the spatial and the temporal variability in strontium isotope ratios across the basin. And you can see a pictorial representation of this model here. It's a Bayesian dendritic model for those who are interested in models. And what this shows is quite good distinction between the northern and the southern basin with regards to water strontium isotope ratios. Also along the River Murray and the tributaries of the River Murray and the tributaries of the northern basin. So this is the map which we use to cross-reference our fish otoliths with. So the second part of the EUCA project was to characterise golden perch populations across the Murray-Darling Basin. And with our research partners, we were able to collect samples of golden perch populations from 14 rivers or regions. And this is a representation of those regions. And the first thing we did was we took an otolith out of those fish and we looked at the age structure of those golden perch from those regions because we were interested in relating flow to recruitment. The second thing we did was we took another section from those otoliths, and colleagues at Melbourne Uni and the University of Western Australia used laser ablation to essentially blast a little bit of the otolith from the core to the edge to look at strontium ratios across the fish's otolith. And this plot here on the left-hand side is a representation of one of those transects of otolith strontium. What we've got on the plot is a black line, which shows otolith strontium from the core of the otolith to the edge. And we've also got these dashed lines here, which are the annual increments on the otolith, so we can look at age. So this was an 8-year-old golden perch. And we can see otolith strontium vary from the core of the otolith to the outer edge. And what we used to do was look at these otolith strontium values and essentially try and cross-check them with our isoscape. But through the EUCA project, again, Ben Fanson, who's just a brilliant guy, a brilliant sort of mathematician, I suppose, and understands biology, Ben developed an algorithm that would match otolith strontium with the isoscape in a mathematical sense. So it would look at each point along this transect and predict with an associated probability where that fish was at that point in life. And this was a very novel approach. You can see here it gave us a pictorial representation of where the fish was caught and its predicted origin, and also some probabilities over here. So this fish was born in the mid to lower Darling River and as a 1-year-old, it transitioned to the Lower Murray and spent a good deal of its life there, and then as an adult transitioned back to the Lower Darling. And what we were interested in the first phase of the Euchre project was associating recruitment in early life with flow. What we also noticed though, that there was substantial movement of fish throughout their lives, and that's what we've become interested in for the MDB project. This plot here is again our samples of fish from each of the regions, and it shows that in some regions, movement from other areas is a strong driver of population structure. So for instance, in the Lower Darling here, most of the fish that we caught were native, in the Lower Darling, were native to that region. They were born there and they remained there for a lot of their life, similar in the Condamine Ballon. But in the Lower Murray and the Mid Murray, we have a lot of fish there that were actually migrants from other regions. And this is another way of actually looking at that. So here's a plot of fish from the Moonee River, about individual transects from 40 or 50 fish from the Moonee, and the same from the Mid-Murray. What we see in the Moonee is that the otolith strontium is stable for a lot of those fish's lives. So they were born in that region and they remained in that region. Whereas for the Mid-Murray, you can see a range of origins of those fish and then considerable movement throughout their life to end up in the Mid-Murray. So a much noisier plot, migration is more important. So if we come back to an individual transect again, I'll describe what we're interested in the MDB project. So this is a 12-year-old golden perch that was born in the Mid-Murray in about 2005, 2006. So it was born in association with the spring pulse, something that a lot of people are talking about at the moment for this year. That fish transitioned into the Lower Murray very early in its life as an early stage juvenile, then spent 4 or 5 years of its life in the Lower Murray before as a 6-year-old transitioning back to the Mid-Murray, probably in association with higher flows in 2011, 2012. So what we're interested in is looking at what flows were actually associated with these movements, both early in life and also later in life as an adult. This is for one individual fish, but for the MDB project, we had this sample of about 800 golden perch from across the basin, so we can aggregate all that data. I've been talking primarily about golden perch today, but we're also going to be doing the same for Murray cod. And when we aggregate all that data, can we see consistent patterns in the flows that drive movement of early stage juveniles or juveniles and adolescents or adults? Our analysis, just quickly, is going to be quite similar to that described by Jason for the acoustic data, but we're going to be using an annual time step because we're using years of data for these fish rather than an event base. We're going to be looking at developing probabilistic models for juveniles, potentially adolescents and also adults, so relating flow to the probability of movement occurring. We've got the potential to look basin-wide at this, or we can break it down into regions or subcatchments. And we can look at contrast between species, so that's golden perch and Murray cod. Just quickly, I'll finish up. How will this knowledge help flow management? Well, all of us who are involved in this project are working very closely at the moment with flow managers across the basin, looking at movement. And so we're sharing this knowledge as it occurs. Jason's will enable the first part of the project us to predict movement responses to site scale watering events, even in unmonitored regions. The second part of this project will enable us to predict flow-related movements that potentially influence population structure. So as I said before, dispersal of juvenile fish, migration of adult fish. Also, this knowledge will feed directly into the fish population models, which Charles is going to talk about in just a second. And ultimately, we can look at integrating the work we did in Euchre with this work, potentially to develop multi-year hydrographs that look at recruitment and movement to promote fish population growth. And we'd also like to be linking with the productivity and other components of MER. All right, I will leave it there and let you get into it, Charles. Thank you.
Thanks, Brenton. If you can just keep your visual up just for a moment until I get my screen up, if you can verify it for me. How are you looking there, Brenton?
Can't see your screen yet.
Okay.
I reckon we're just about to see it. Yep.
Great. Thanks, Brenton. So good morning, everyone. I'm Charles Todd, and I'm developing a number of fish population models with the objective to understand the contribution of chew water to native fish population processes. So, and this is the program outlined over the next few years. The F1 fish population models program is to deliver Murray-Cod selected areas model and meta-population models for basin-wide species of golden perch and bony herring. And Murray-Cod and golden perch are obvious species to focus on, but bony herring perhaps less obvious. And the reason for our interest in bony herring is that they are a flow responder and an important prey species throughout the basin. So the bony herring model will be developed at a basin scale similar to golden perch, and the schematic on the left there is a representation of the golden perch metapopulation model and the spatial scale that it will cover. So what is a population model? So a population model takes the life history of the species that we're interested in and converts that life history into a series of mathematical relationships. And so we undertake a life history analysis of the species and then develop that into a population model. And in our case, it'll include fish field data and hydrology. So the population model is to develop to predict population scale response to flow management and can be used to identify optimal strategies and outcomes. So flow influences many or all of the life history elements for fish in our river systems. And part of that influence is movement, and from which we'll be drawing on knowledge from the MER F2 fish movement program that that Brenton has been speaking about. And then and these models can be single species or multiple species. So developing fish models to look at the influence of flow on fish population dynamics is a highly cost-effective and it is highly collaborative as well in the way that the information is collated. And they also help identify key questions, and where required, we can make transparent assumptions. So this approach is scientifically defensible for making evidence-based decisions for ecological outcomes and can be validated. So here's a schematic of how we take a life history model and apply it to a variety of different locations. So the life history model can be applied to multiple locations across the basin. And for a metapopulation model, movement, immigration and emigration is defined between each population. And most movement is driven by flow, both upstream and downstream. And analysis of flow data is used to drive these connections between the populations. And again, we'll be drawing on the knowledge and insights from the MUR F2 fish movement program to help define these movement flow relationships. So once these complex models have been constructed, different flow scenarios can be examined for their effect on population response, and these outcomes can be ranked. And so the life history of golden perch, large-scale movement that they undertake, and the potential significant spawning that can occur in nursery sites requires a coordinated basin scale management. So here's a conceptual example of the kind of things that we expect to be able to do. And so we could, for example, model the natural population response. We have access to flow data of the expected natural flow, and then we can look at potentially multiple interventions, the chew watering alone, and then potentially no intervention. And so we would expect that these different hydrological examples would provide different outcomes in the population model, from the population model. And then this would enable managers to choose their preferred population trajectory, and this decision can be done in an open and transparent manner. So here we have a real world example of some recent work, and I'd like to acknowledge VEFMAP and thank them for allowing us to represent this work here. So we have a Murray cod model for the Campaspe River, and we have here two different flow scenarios. And so the dark line being the actual flow scenario and the light blue line being the current e-water recommendations. So the model predicts Murray cod population trajectories for a range of flow scenarios, which we've presented to here. And it's built on real field data, daily hydrological, fish surveys, and statistical analysis of key metrics that influence spawning success. So in this scenario, the current EWA recommendations are superior to the actual flow, and the model predictions are corroborated by long-term field data. On the right-hand side, we have an associated risk curve, and the dark blue line being closer to zero indicates that this risk, the actual flow has a higher risk of lower population size than the light blue risk curve associated with the current e-water recs. And so we can test a range of scenarios and interventions and then rank them based upon their risk outcomes. Validation can be incorporated, and again enables managers, communities to make informed choices concerning their objectives and investments. So it's a highly efficient way of setting targets and making fish decisions. And again, the data behind this is the VEFMAP data for adult cod collected over 15 years and a 15-year population model. So the next steps are to develop the strong linkages to the F2 fish movement program, food webs, hydrology, selected area themes, and also to other programs such as VEFMAP, and then provide selected area and basin scale modelling to answer the questions. The question, what did true water contribute to native fish populations? So I'd like to conclude that that fish research enables improved water delivery for ecosystem outcomes. And so the panel will field any questions that our audience has. Thank you.